Hello and welcome to Science Talk, the podcast where we talk about the latest things happening in the science world. I'm your host, Edward Smith, and today I'm here with biologist Chris Pierre, who is the leading researcher at Harvard University in Cambridge. Big stuff. How about you introduce yourself, Chris?
Thank you, Edward. I'm Chris Pierre, and I study germline genetic modification at Harvard. My research team works with embryos and experiments with different techniques of altering the human genome.
Wow, that's fancy stuff. Can you explain a bit more detail what germline genetic modification means? That's a lot of big words, and I'm not sure I got them all.
Yeah, sure. All the cells in your body fall into two categories. There are somatic cells, which are most of the cells in your body. Then there are reproductive cells like ova, zygotes, oocytes. Germline genome editing, which I'll call GGE for the sake of efficiency, it targets the reproductive cells in your body. If you were to target the somatic cells, the changes wouldn't be passed on to your children. But with GGE, the changes may stick and will be passed on to your kids, your grandkids, etc.
Wow, okay, so you could change my eye color and then all of my future generations would have that eye color?
Yeah, you've got the right idea. The technology is far from being used for aesthetic purposes, though.
Why is that? Does it not work for changing our appearance?
Oh, it could work. The research isn't really concerned with that right now, though. We're primarily looking at how we can use technologies like CRISPR to cure genetic diseases.
Oh, yeah, that sounds a lot more pressing.
Yeah, definitely. My lab is working on altering the Alzheimer's gene in human embryos. CRISPR has worked on Huntington's disease patients before.
That's great news. If it worked on Huntington's, why have we not got it to work for everything else yet?
Well, it's a bit more complicated than that. CRISPR technology works by using this little protein called Cas9. Think of Cas9 as a pair of scissors. You tell the protein where in the genome it needs to target, and it runs in there and starts cutting the genome up. After it makes the cuts, we insert the DNA sequence we want in there, and the sequence binds into its new spot. The problem is being able to target the gene. We were able to send Cas9 to the Huntington's gene, but it still hasn't happened for the Alzheimer's gene yet. We are really close, though. This technology has great potential.
That's really cool.
Cooler than designer babies?
I don't know about that one, but it's still pretty awesome we can do things like that. There was one thing you said about your research that I want to talk about. You mentioned experimenting on human embryos, like the things that become babies?
Yeah, the things that become babies. My lab doesn't use viable human embryos, though. None of the embryos could have ever come to term.
Then do they work? How do we know if it's working if they're not viable?
Yeah, that's a prominent issue in the field. The answer is we don't really know until we try on a human. We can see how the embryo reacts, but we already know this produces variable results. It'd be super helpful if we could use variable embryos.
Well, then why can't you?
Well, there are some ethical issues that have not been resolved yet. The concern is that we are modifying a living thing, and it can be a bit unnerving to a lot of people. Everyone's like, oh, these researchers are playing God, and we can't have that. There are also thoughts that it can be dangerous.
Wow, fascinating stuff. How do you feel about it? Like, would you rather have the viable embryos?
For sure. I don't want to downplay ethical concerns, but I also want to acknowledge the potential for this technology. Currently, risk is fairly low and reward is very high. I'd kill for viable embryos, but they're just too expensive right now.
Well, what makes them so expensive?
Well, the issue is making sure they're ethically sourced. They're easy to find, sure. There are plenty of women out there. But how do we get those embryos? We can't create new embryos because the government bans the creation of embryos for research purposes. The embryos that I use are from leftover in vitro fertilization. So when a couple decides to do IVF, there are embryos that were not viable and thus not used. It's a pretty big hurdle to jump, and only the best funded labs can have access to such things.
Interesting. I didn't realize there would be problems like this in this kind of field.
Oh, we deal with things like this a lot.
Well, you mentioned the idea of playing God. Do you feel like you're doing that? I mean, does changing the genome make you uncomfortable at all?
Not really, no. I obviously think regulations should be imposed. I'm a scientist, but I'm not a monster. I want GGE research to be safe, but I also want it to be effective. The thing that bothers me is when people are like, you don't know what's going to happen. Like, yeah, that's why I'm trying to find out. No matter what research you're doing, you're jumping into a whole new world. That's how you discover things. Of course, I condemn careless experiments, but I could never say that we shouldn't research something just because we aren't fully sure what the results will be.
Wow, that's really inspiring, Chris. Unfortunately, that's all we got time for today, folks. If you want to hear some more about Dr. Pierre's website, you can visit it at chrispierre.com. Thank you so much for your time here today, Chris.
Yeah, it was a pleasure.
After this quick ad break, stay tuned to hear us talk to Dr. Honey Busby about the overlooming bee extinction crisis.